Hello everyone, today we are going to talk about cardiovascular physiology. Within our heart, we have two types of myocardial cells. We've got autorhythmic cells, or you can call them pacemaker cells, and then the other type is myocardial contractile cells. Contractile cells are the ones that are going to contract so that your heart would push the blood out of its chambers. The autorhythmic cells, those are the ones you see right here. Those autorhythmic cells are not going to contract. They are going to send electrical signals or action potentials that are going to spread throughout your heart. Um, our autorhythmic cells, we have about 1%, they make up about 1% of your cardiac cells. The rest are going to be the contractile cells. Those autorhythmic, auto meaning self, and the reason why we call them self, because they can initiate their own action potential spontaneously in the absence of any input from your nervous system. Once those autorhythmic cells generate their own action potential, the action potential is going to spread throughout the heart, and it's going to spread to the contractile cells. Once the contractile cells receive the electrical message from the autorhythmic cells, those contractile cells are going to generate their own action potential, and then following that action potential, the heart is going to contract. So you will notice that the electrical signal always precedes the mechanical signal. And those electrical signals are going to originate in the autorhythmic cells first. Then the message is going to be transmitted to the contractile cells. Then the heart can go ahead and contract. The autorhythmic cells that we have, we've got the SA node, AV node, the AV bundle, the bundle branches, and the Purkinje fibers. SA stands for sinoatrial node. Atrial because it is going to be in the right atrium. That's why it's called the sinoatrial node. The message, the electrical message, is going to be initiated within our SA node. SA node takes the lead. Then the message gets sent to the AV node. Then it's going to be sent into the AV bundle, then into the bundle branches, and then into the Purkinje fibers. The message is also those electrical messages that are going to be initiated by those autorhythmic cells. They're also going to spread throughout the heart chambers. And as you can see right here, the purple color is representing the action potentials or the depolarization wave spreading throughout your heart chambers. And as I said, those electrical messages are going to spread to the contractile cells so that the contractile cells can generate their own action potential, and then following that, the heart would start to contract. Electrical message always precedes, as I said, the mechanical message. Now, our SA node, why is it the one taking the lead? Because it is the strongest of all of the autorhythmic cells we have, and it's the one that's going to set your heartbeat to 70 beats per minute. Now, if your SA node, which is the one taking the lead, if something goes wrong and it fails, then who's next? The AV node. So the AV node will take the lead, and it's going to set the pace of your heartbeat to around 50 beats per minute. As you can see, it's slower compared to the SA node. What if your AV node fails? Then the Purkinje fibers are going to take over, and those are very slow. Those are going to set your heart rate to 25 to 40 beats per minute. And so it's really very hard to survive on your Purkinje fibers because your heart rate is going to be extremely slow. And that's why you hear individuals getting a pacemaker. The reason those individuals are getting pacemakers, because it could be that their SA node, which is, as I said, the one in charge, has failed, and so they need a pacemaker to be able to have a regular heartbeat of around 70 beats per minute. Now, as you can see right here, the SA node is going to initiate an action potential, and that depolarization wave of the action potential, as you can see, it spreads throughout the atria. Now, when it spreads throughout the atria, who's going to pick up those electrical messages? Your contractile cells. Following that, the contractile cells will initiate their own action potential, and then your atria is going to start to contract. Now, in the meantime, as this is happening, the message is also being sent down to your AV node. But when the message gets sent down to the AV node, you're going to notice that there's going to be a slight delay in the transmission of the action potential once it reaches the AV node. Why is there a delay of the action potential once it reaches the AV node? So that you can allow time for your atria to contract. So you don't want to send those electrical messages quickly down into the ventricles, because as I said, the mechanical message follows the electrical message. So once the electrical message goes down the ventricles, eventually the ventricles are going to contract. And you don't want your ventricles to start contracting if your atria have not even finished their own contraction. Because when your atria contract, they're going to push the blood into the ventricles. And so if your atria are not done pushing the blood into the ventricles and the ventricles started contracting already, then you're not going to be pushing enough blood out of your heart. And that's why the electrical message, once it reaches the AV node, there's going to be a bit of delay before the message goes down the AV bundle, as I said one more time, so that your atria would have enough time to contract and push the blood into the ventricles. As we said, the autorhythmic cells, or you can call them the pacemaker cells, they are going to generate action potentials spontaneously, right? Meaning they don't need any input from your nervous system for them to go ahead and initiate those action potentials. And once the action potentials originate in your pacemaker cells, those action potentials are going to spread to the contractile cells. How are they going to spread to the contractile cells? Via the gap junctions in your heart tissue. Now, right here, we are going to explain the steps of the action potential initiating in your autorhythmic cells. Now, the condition in your autorhythmic cells, the pacemaker potential, it's around negative 60 millivolts. Now, you can think of the pacemaker potential kind of like your resting membrane potential, but in your heart, we refer to it as the pacemaker potential because some say that it could actually differ. It's not always set exactly to be negative 60 millivolts. Now, for an action potential to be initiated, Remember how we talked in our action potential video previously that you must reach a threshold. After you reach a threshold, an action potential will be initiated. So as you can see right here, we are down at negative 60 millivolts. We have to reach a threshold that's around negative 40 millivolts. So how is your autorhythmic cells going to reach the threshold of negative 40 millivolts? Two channels are going to play a role in reaching that threshold. Those channels, one of them is known as the IF channel, and the other one is calcium channels. IF, F is actually stands for funny. So those are called the funny channels. And the reason why they call them the funny channels is because when they first discovered those channels, they thought that those channels are kind of acting weird, and so they called them funny channels. What are those funny channels going to do? Well, those funny channels are going to allow sodium influx and potassium efflux. Influx meaning the sodium ions are going to be entering the cell. Potassium efflux meaning that potassium is going to leave the cell. Now, sodium is positively charged. So if you have a bunch of sodium ions entering into the cell, what's going to happen? The cell is going to become more positive. But some are going to say, well, potassium is also positive and potassium is leaving the cell. The thing is, those funny channels are going to allow more sodium to enter into the cell versus potassium leaving the cell. And so because the sodium influx exceeds the potassium efflux, you are going to see that depolarization is going to be initiated. But it's not going to allow that depolarization is not strong enough to reach threshold on its own. And so another channel that's going to help reach that threshold is a bunch of calcium channels. Those calcium channels are going to open up, and calcium, which is also carrying a positive 2 charge, the calcium ions are going to start entering into the cell, causing more depolarization. And so as you can see, the depolarization wave is eventually going to reach threshold. Once a threshold is reached, then an action potential is going to go ahead and begin. So for us to reach threshold in those autorhythmic cells, again, two channels play the role, those funny channels and the calcium channels. Now, right here, we've reached negative 40, so an action potential is going to begin. A bunch of calcium channels, voltage-gated calcium channels, are going to open. And by the way, those voltage-gated channels that are going to open right here are different than the calcium channels that opened down here, okay? So a bunch of voltage-gated calcium channels are going to open. More calcium is going to start entering into the cell, causing a steep, right here you can see that steep depolarization wave. Now, once we reach right here a positive voltage, like around 20 millivolts or a bit less actually, the calcium channels are going to close and potassium channels are going to open. Now, once potassium channels open, what's going to happen is potassium is going to start leaving the cell. The more potassium leave the cell, as you can see, the wave starts to drop because we are going back, we are trying to go back to the pacemaker potential. And so as you can see, repolarization is going to take place because potassium ions are leaving the cell. And then once we hit the pacemaker potential, those potassium channels are going to close, and you can see um, another cycle is going to begin. So this right here, those are the steps that are taking place within our pacemaker cells. Once this takes place in our pacemaker cell, this action potential right here is going to be transmitted to the other cells in your heart, and they are going to be received by your contractile cells. Now, till now, your heart has not contracted yet, right? Because as we said, the electrical message precedes the mechanical one, and those autorhythmic cells cannot initiate contraction yet. They will send those action potentials down to the contractile cells, and it's going to be sent via the gap junctions. The contractile cells are going to initiate another action potential, and then your heart will go ahead and start contracting. So as you can see right here, those autorhythmic cells, like your SA node, for example, they are going to go through that action potential that we just discussed together, and those electrical signals are going to pass through the gap junctions that you have to the contractile cells. Once the contractile cells receive those electrical messages, they are going to initiate their own action potential. And then the muscle is going to start contracting. Our myocardial contractile cells have a resting membrane potential of negative 90 millivolts. When a wave of depolarization moves in from the autorhythmic cells into our contractile cells via the gap junctions, the membrane potential of our contractile cells will start to become less negative. That's because a bunch of voltage-gated sodium channels are going to open up, allowing sodium to start entering into the cell, depolarizing it even further more until it reaches about positive 20 millivolts. That's when the sodium channels are going to close. Then a bunch of potassium channels, we refer to them as fast potassium channels, are going to open, where potassium is going to start to leave the cell, causing repolarization. So as you can see right here, the graph is dipping downwards because repolarization is beginning. But notice how the repolarization does not last long. It's very brief, and we get that plateau phase right there. So what's happening is that some of those potassium channels start to close. And so now you don't have a lot of potassium leaving the cell. And at the same time, we have voltage-gated calcium channels that open. And calcium starts to enter the cell at the same time as the potassium is leaving. So let's think about it. Calcium is positive charged, and potassium is also carrying a positive charge. If you have some positive charged potassium ions leaving the cell, and at the same time you have positive charged calcium ions entering the cell, you are not going to have a full repolarization or a full depolarization. You get a plateau because you have positive charges entering and positive charges exiting at the same time. So we get that plateau phase right there. Then what happens is at the end of that plateau phase, to end that plateau phase, the calcium channels are going to close. So if the calcium channels close, you don't have any more calcium entering the cell, and at the same time, potassium channels open, and the ones opening right here, we refer to them as the slow potassium channels. So now you have no more calcium entering the cell, and you have a bunch of potassium ions leaving the cell, which is going to cause the graph to start dipping down, and you're going to have repolarization. And that repolarization, potassium ions are going to keep exiting the cell until we go back to the resting membrane potential. So those are the steps of an action potential taking place within our contractile cells. This image right here is comparing the action potentials and muscle contraction in your contractile cardiac muscle cells versus your skeletal muscle cells. The red graph is representing the action potential. The blue graph is representing the muscle contracting and relaxing, and the yellow highlighted region is representing a refractory period. A refractory period is a time following an action potential during which a normal stimulus cannot trigger a second action potential. Now, one thing to note between the action potential of our contractile cells versus the action potential of our skeletal muscles is that the action potential of our contractile cells lasts longer, about 200 milliseconds, versus in our skeletal muscles, the action potential, it's way shorter. It only lasts from 1 to 5 milliseconds. Now, why is there such a big difference? And the reason is, in our contractile cardiac muscle cells, we hit that plateau phase during our action potential. And that plateau phase right there is what causes the action potential to stay longer. So you don't have a quick repolarization in our cardiac contractile cells. Since our skeletal muscles, we don't have that plateau phase. Once your action potential, you get that steep depolarization, we are going to open the potassium channels and we're going to get that quick repolarization. Why is having a longer action potential in our cardiac muscles pretty important? Because that's going to prevent sustained contractions from taking place within our heart muscles, known as tetanus. Preventing tetanus in our heart, and as you can see right here, because having a longer action potential is also going to lead to a longer refractory period. We are not going to have several muscle contractions happening at the same time. You can see right here, the muscle is contracting and then relaxing, and then it starts to contract again and relax. Versus in your skeletal muscles, because the refractory period and the action potential is very short, if you keep stimulating that skeletal muscle, you can have summation of the muscle contractions to the point that the muscle is not fully relaxing before it hits with another contraction. And you get that summation of contractions, and you get what we refer to as tetanus. Now, preventing tetanus in our cardiac muscles, it's very important because your cardiac muscle cells must relax between contractions. They must relax between contractions so that we can give time for our ventricles to actually get filled with blood. Your ventricles, when they are relaxing, that's when they get filled with blood. So if your muscle cells are constantly contracting and you're not even giving it time to relax, then your ventricles are not going to get fully filled up with blood. And so therefore, if the ventricles contract right away, they are going to be contracting and pushing a very little amount of blood because you didn't give them a time to relax, meaning you did not give them time to get filled with blood. The action potential of our myocardial contractile cells will move down the surface of our cells where we have our sarcolemma. That action potential is going to move down into the T tubules, and this is where the events of the excitation contraction coupling will go ahead and begin. As the action potential makes its way into the T tubules, we have voltage gated calcium channels. Those channels are going to open, where calcium is going to start flooding and entering into the cell. That calcium entering into the cell is going to cause or it's going to lead to the opening of receptors on the surface of the sarcoplasmic reticulum known as ryanodine receptors. Once ryanodine receptors open up, the calcium stored within our sarcoplasmic reticulum will be released out. So what causes ryanodine receptors to open is calcium. So that's why we refer to it as calcium is inducing the calcium in your sarcoplasmic reticulum to be released. And once calcium is released, calcium is going to bind to the troponin protein found on the actin filaments. Once calcium binds to troponin, the cross-bridge cycle can initiate. And once the cross-bridge cycle is initiated, the heart muscles are going to start contracting and eventually also relaxing. Now, the events of the excitation contraction coupling in our contractile cells is different compared to what's happening in our skeletal muscle cells. But the cross-bridge cycle is exactly the same. So the cross-bridge cycle in our skeletal muscles is going to be exactly the same as the cross-bridge cycle in our cardiac contractile cells. For our cardiac contractile cells to relax, the calcium is going to unbind from troponin, and the calcium is either going to get pumped back into the sarcoplasmic reticulum or it's going to be released out of the cell. And actually, both of them can happen at the same time. So the calcium is going to be pumped back into the sarcoplasmic reticulum via the help of the calcium ATPase pump. And at the same time, some of the calcium can actually be released out of the cell via a pump known as the sodium calcium exchanger. And the reason why it's called exchanger, because as one calcium is released out of the cell, 3 sodium are going to enter the cell. Now, the 3 sodium ions that enter the cell, they're not going to stay for a very long time because they will be pushed back out via the sodium-potassium pump, pushing 3 sodium out and 2 potassium in.